Okay, welcome everybody. We are here to listen to Dante Chialvo. He will use his brain to explain to us what what happens in our brains, and it's interesting because he studied medicine, but he is using statistical physics, and he's a fellow of the American Physical Society. So he will use all his experience to explain this to us. Please, Dante, you're welcome.
Okay. Thank you, Alicia, for the invitation, and thank you all of you for for having me today. I don't know if with the title of the talk and the abstract, I have raised the expectation well above of what today I will be able to cover. But as I said in the abstract, the question that we are interested is a very simple, but at the same time very fundamental one. When you observe nature, you see that life at large is extremely complex, and and so the question is, where is coming all this complexity? What is the physics beyond such complexity? And I have written and below actually sort of the moral of the story that all the physics laws are simple, relatively simple. The most fundamental one don't have more than 3 or 4 letters. But nature is complex. So the question is, do we need to have complex laws to explain nature, or simple laws will explain everything at once, uh, providing that you are in the right region of parameter space? That's all. Um, as As I said, we have worked in different domains of nature, from social insect to single molecules to proteins to entire brain, small brains, small regions of the brain, etc. And in all the cases, we don't do theory ad hoc. We use the same fundamental principle, which I try to explain today. And I guess that the main message should get through. Otherwise, you interrupt me at your own risk and ask for clarifications, or you can go to the website and see if some of the papers that are in my website can clarify something. The outline is this. I will mumble around why complexity often equals criticality. I will define what criticality is, and um, vis-a-vis complexity. I will briefly comment on what is the critical brain hypothesis that we've been working for a number of years. Then I will get into the data and show the most relevant evidence so far that criticality and complexity in the case of the brain seems to be a useful approach. I will get into the concept of scaling, which for mathematician, I don't need any clarification, but in the case of the physics of these processes, there is a particular one that links space and time, that is called dynamic scaling. And then, as a curiosity, I will describe how we have suggested that criticality can be controlled, in the sense that not to avoid it, but to make the system stay critical all the time. Then there is some time I will some remarks. This is the list of some of the papers that you can find in the literature that we have um, discussed these issues. The red are reviews, the blue are the most recent ones dealing with different systems. In this case, I think that all of the blue ones are concerning the brain. Again, this will be in the slide, so you don't need to. If you are interested, you can go and read it. Okay, so what are the phenomena that you are dealing with? Okay, you have here 2 scales. One is the my brain speed 7 times. And what is red or yellow is the region of the brain that been more active, and the blue one that were more inactive. Okay, this is the amplitude of the consumption of oxygen in the brain, which with technique of magnetic resonance can be recorded. At the right side, you have the brain of a fly, which is make it fluorescence when the voltage of these neurons, these spots, are individual neurons. As you can see, you have flashes that suddenly cover the entire brain, and it's extremely regular, more or less the same that the reality that you see at the large scale. I have these flashes, these avalanches that are typical of the spontaneous dynamic of these brains. And the next sample is even a scale of much smaller. You see, these are 10 microns, the scale on the bottom, sorry. It shows that these are the activity at the individual synapses connecting several neurons, because you see there that the intersection of the axons and the dendritic trees. And this is the typical phenomenon. Okay, so that's the problem you have to solve. Where is coming these dynamics? How you can describe that type of dynamics? And we have, for a number of years, suggested that these are examples of complex phenomena that all of them, independent of the scale, are generic of critical phenomena. Critical phenomena in physics refer to the phenomena they observe in a phase transition between two states. In an extended system, a large distributed system, it could be the prototypical example is the Ising model, for instance, for magnetization, in which individual degrees of freedoms are the spins, and these spins are coupled with the neighbors in ferromagnetic way, and they try to order it. There is a temperature that agitates the spins, and there is a collective process that is fighting against, and depending on the temperature, the system is completely disordered, or the system is completely ordered, it is frozen in one of the configurations. But at the critical temperature, was so-called for that situation the Curie temperature, because it was described by the Curie in more than 100 years ago. At the critical temperature, you have that is a mix of order and disorder. Okay, these phase transitions have changed the way in which we understand nature, because before um, it, it was not conceivable that something like ice can become liquid with a difference of 1 degree temperature. Alchemists were thinking that ice and liquid water were different matters, and for that reason they were trying to melt lead and get gold, because in their mindset these were different matters. And indeed, when phase transitions were described, thermodynamics start, and the way in which we understood nature was completely different and evolved through the last 2 centuries to the concept that we have today, that there is a structure, there are interactions, and depending on the single parameter, could be temperature, pressure, or volume, you can get the completely different phases with a completely different configuration and appearance. But if you are at the critical point, for the last 40 years, people have been wondering if what we call complex was just an expression of being near this criticality, in which the two states, the order and the disorder, um, um, are spontaneously mixed. Hans Frauenfelder, he died a few years ago, worked until 3 days before he's 100 years old. He's in that picture. I was a physicist working in the last few years in Los Alamos. And this is a picture, a cartoon that he published in 87 when I met him at Los Alamos, that strike me as very, very meaningful. You have a qualitative axis for complexity, a horizontal axis for low temperature or high temperature, order and disorder. And he said that complexity maximized in the intermediate point between order and disorder. And he wrote life, brain, languages, proteins, turbulence, and glasses. So he was so advanced in his time that basically predict the next 30 years of discovery in biology, because now we know that all these concepts are associated with phase transition. In the 90s, this person with the blue pullover, Per Bak, advanced a theory. The name was self-organized criticality because he provided a toy model in which energy being pumped in all these degrees of freedom of any system in particular. The system was non-linear in the individual degrees of freedom, and this energy being dissipated through avalanches. In this avalanche will make the system subcritical, but the energy being provided will make the system reach criticality again, and so on and so on. He studied this toy model and described many of the phenomena that occur in nature and start associating the complexity that you see in nature, quantitatively speaking, scaling laws and rigorous descriptions, and popularize the concept of criticality through biology, economics, social sciences, so that today the experiments that people have conducted demonstrate that indeed physical, social, biological systems are shown to be complex because they operate near criticality. So it seems that all these 4 decades have facilitated the job of scientists in the description of complex phenomena, in the sense that there is a mechanism which is extremely robust, that is universal, that will, as soon as the system get near criticality, it will start expanding its complexity with certain quantitative prediction that one can make. This is the manifesto that I mentioned before, and in this manifesto, what we suggested was that the problem in the brain, the most fundamental problem is how the brain managed to produce all the cortical configuration, all the possible combination of activity, but in a flexible manner, not as a circuit. The brain cannot be a circuit, obviously, because as I speak, I am changing my configuration. I will need another brain in order to change the connections if my brain were a circuit. Another circuit, so we suggested that a possibility was that this mechanism that exists in dynamical system, a very large dynamical system, that are near a second order phase transition, can be the reason or the mechanism for creating and reconfiguring in this region of marginal stability that is typical of criticality. Okay, so I don't have too much time to explain all the rationales of the hypothesis. I prefer to show you what is the evidence that looks like that. Okay. Questions so far? I don't see any face, so I am talking to
Yeah,
dark
I
matter.
forgot to
So
say that you can ask questions while he's speaking, and he will do his best
yeah,
to
yeah, it will be
not to
good if there
deceive
is at
you.
least one face in the screen, so that if there is anything I can anticipate. So as I said, if there's no question, I will continue. I realize that I'm not explaining to you what critical phenomena is. I'm hoping that as a scientist, at least you know this concept marginally. This slide will give you the examples that are most relevant in critical phenomena as you see it in the brain, but if you change the brain for another system, even a ferromagnet at the Curie temperature, this property will be there. The title says relevant universal correlation property of critical phenomena demonstrated in the brain. Critical phenomena is described by the properties of the correlation function. Basically, the business of critical phenomena is to calculate a correlation function and see in which universality class this object that present a critical phenomena belong. The typical, the most general properties that you will see that as you approach the critical point between two phases, you will have the largest variability, estimated in various ways. It means that if you repeat the experiment, you're near the critical temperature, the configuration, the variance of your observable will be maximum. Okay. The second one is that in space, you will see clusters of all sizes. In space, in physical space. If you look at the sky, your cluster will be the size of the clouds at any time in the day, average over the entire year. If the atmosphere is critical, you will have a power law distribution of cluster size, and that was been proven many times after Per Bak raised this issue, showing that indeed you can have the largest cloud will cover the entire planet, okay, with probability 1, and then a smaller cluster will have probability 1,000, and in the middle you have a power law 1 over x. Okay, so the third thing that is extremely useful is that if you take the second largest cluster, not the first largest, but the second largest cluster, the size of the second largest will peak at criticality, because as soon as you move your control parameter, the atmosphere get hotter or colder in thermodynamic sense, the second cluster will be either coalesce with the largest cluster or will disappear. So you have a competition between all these clusters that is guided by the correlation, the underlying correlation of the system. There is no magic there. All these things are tied together. So if you go and do an experiment in the brain, which this is a 2012 paper of a undergrad student in the University of Buenos Aires, the physics department, Enzo Tagliasucci, was a student of mine at that time. What he did is to put a subject in the scanner, in the fMRI scanner, magnetic resonance imaging scanner, and take the data like I showed you before in my brain at seven times speed at the beginning of my talk. And then after massaging the data a little bit to identify what clusters are, that I don't have time to go into detail, but basically is to pick in the highest density of activity, putting dots there, convert the entire process in a point process, and then start clustering the spatiotemporal point process, and then defining clusters, counting the clusters, panel C, what it says here, largest variability. He will plot the number of clusters between 0 and 150, these are the number of clouds in your brain, as a function of the number of active sites, the number of how much activity you have in the brain at that particular moment. And you do this over time and time, and this is your statistic. You will see that the curve goes up and down. To make an analogy, when there is zero cluster and zero activity, there is no cloud in the sky. When there is 10,000 active sites and there is one cluster, it's completely covered by a single cloud. And in the middle, where is this red line, the red arrow, is where you have clouds of all the sizes. On the panel D, in double log, you will see the histogram of the probability density function of all these, the size of these clusters. And you see that it's a nice power law with an exponent of 3 over 2, which, by the way, is an exponent that came from the statistical physics of this phenomena. It's exactly what you get without any fitting. And it's not just a single experiment. These are overimpose 4 or 5 experiments with no normalization, you get exactly this 3/2. Furthermore, if you now cluster your data, go to panel F, if you cluster your data for low activity, high activity, and middle activity, you will see that the blue one is low, kind of fall off exponentially, while the red, which is kind of higher activity, too much activity, you have this extra bump on the 10 to the 4, which is like these are the big avalanches of activity, exactly like the theory predict. If the system is switching to the left of criticality, to the right of criticality, in and out of criticality. If you put all the data together, you get panel E. In panel E, the data is now replotted considering defining an order parameter. The order parameter is the size of the largest cluster, which I mentioned before, which is a very useful definition to keep track of how ordered the system is. So the green points are all the data. The red is the average of the green points, and you see that you have a sort of sigmoidal function, and that is typical of a second order continuous phase transition in other systems. But the fact that you have a sigmoidal function is not sufficient to say that you have a phase transition. What you have to have is that at what you think that is the phase transition, which is the red line, because I'm using as a reference the peak of variability at the clusters, you see that at that line, the variance of the data is larger, which are the black squares. So that means that indeed, when you reach this intermediate critical point, the variance increase as is predicted and expected from criticality. But then you say, Dante, but you have a logarithmic axis that goes from 100 to 10,000. So is the system critical or is in and out of criticality? It's in and out of criticality. As you expect, the atmosphere is never at the critical point, but is switching in and out. This is telling you that indeed something of the mechanism that you are suspecting is present. Nevertheless, if you compute the resident time, this is the blue line over here in panel E, you will see that most of the time, okay, the system is near the critical point. The most of the time is critical, and of course it's wandering in and out. It seems that the forces that regulate these things in the entire brain are counterbalancing each other. There is a number of explanations for that, such that it always get back to criticality. So all these observations are extremely suggestive that indeed the brain at the scale of the entire human brain is critical. And then another student of another licenciatura in the same department in Buenos Aires, Jaimovici, he did his PhD thesis and his licenciatura studying a model which have a single parameter, which is just the control parameter, is like a temperature, a canonical temperature, but it's based on the connectivity of the human brain. So he took the connectivity of the human brain at large, coarse grain, of course, and changed the threshold, the temperature of the model, and then compare the experimental data with the result of the model that I insist. The model have no fitting parameter. And when the model was at the critical temperature of the model, the agreement between the experimental, this is the diagram on the left, which is the probability distribution of the cluster sizes. You see that the red is the model, and the crosses, the black crosses, are the experimental data. And both of them have this 3/2 universal exponent. The only difference is that the experimental goes one order of magnitude more to the left, to the right, just because it has 40,000 degrees of freedom, while the model have only 1,000 because of the coarse graining necessary to get the data. So these were extremely uh, uh, um, I will say, extremely good result 10 years ago at large scale, which were the only data that was available for us to test these theories. Up to now, I have talked about basically densities, probability distributions, but I insisted that the business of criticality, the undisputed business is correlations. And there is one particular that is the hallmark of critical phenomena. In critical phenomena, if you measure correlation length, which in this audience of mathematicians, I don't need to explain very much, you measure what is the zero crossing of your correlation function. There are different tricks. One that works is to subtract the mean of your fluctuations so that now you have what is called the connected correlation function. And this connected correlation function necessarily cross zero, and you have anti-correlation, positive and negative correlation. But it doesn't matter. What you're looking for is the zero crossing, which is the only distance at which the phenomena, the degrees of freedoms are uncorrelated. And it turned out that at criticality, because of the properties of criticality, there is no correlation length. The correlation length is the correlation length of the system, is the size of the system. So you enlarge the system, the correlation length increases. So there is no finite correlation at the critical point. So to measure this function is extremely relevant to get evidence of critical phenomena. So recently, actually, there are two results that are separated by 10 years. I will describe first the latest one from Daniel Martin at the University of San Martin, Argentina, that he asked the following question. When you measure the correlation length, you have systems of different length. If it's a model, you increase your lattice size, and then you recompute. But in biology, you cannot cut the brain in half or in a quarter or in 8 or 16. So he asked, can I change just my window of observation? And I will get exactly the same if I change the system size as traditionally in critical phenomena. And in this paper a few years ago, he showed that indeed he did it first with the ICID model, which is a prototypical model in this business. And here you have three cases: the ICID model very, very cold, the ICID model very hot on the right, and the ICID model at the Curie temperature. So the configurations that you have at the subcritical is all spins in one direction. The configuration high temperature, all the spin changing all the times, uncorrelated. And the typical situation is the middle one. When you are at the critical temperature, you have islands of spins correlated at any given time, and these islands are a fractal with some distribution with some power law. Well, now you take the 2-point correlation function, you subtract the mean of fluctuation, you compute the correlation, the connected correlation function that I presented before, and you plot it as a function of the distance, you will have several curves. Okay, let's take the subcritical case. You have several curves, each one corresponding to a window of observation larger and larger. And you see that if the temperature is below the critical point, meaning the system is frozen, you have indeed that as you increase the length of your observation window, nothing happened. You have that asymptotically you reach to the interaction length of the spins. Notice that the scale is between 0 and 20. But if you go to higher temperature, okay, to the supercritical, again, you have that as you increase, if you go from the green to the pink, okay, that is going from 16W equals 16W equal 256. You got 2 orders of magnitude change this in the window observation size. Nothing happened. Again, you compute the interaction length, not the correlation length of the system. But if you are the critical temperature, as you go from the green to the pink, you see that the zero crossing move to longer and longer distances. And these distances basically is one half of your square of your window, which is the crossing zero. One domain is anti-correlated with the other domain. This is generic of criticality. If you demonstrate that in a system that your correlation length increase with size, then you are absolutely sure that your system is critical. Furthermore, you can distinguish two kind of increase. At the critical temperature, go to this panel on the right on the top. The top and the bottom are one is linear, the other is log linear. At the critical, okay, you see the green line grows, sorry, at the critical temperature, the correlation length increase linearly with size. You see that below and above that critical temperature, you have a growing that is logarithmic. So you can use this property to know if your system is near criticality, if it's subcritical, it's supercritical. So you go and do an experiment like that, and then you can be sure that things uh, respond to your conjecture theory. This is an experiment done by a paper by Daniel Freiman, again from the University of Buenos Aires at that time. Now it's in San Andres University in Buenos Aires, in Argentina. He computed for the entire brain, for my brain, the correlation length. In the panel on the left, you see all these functions are for different sizes, okay? And the graph on the right is the correlation length as a function of the size of the blob that you are considering in your calculation. And you see that the correlation length increase with the cluster size that you're considering. This figure is wrong 20 years, no, sorry, 14 years after. We should have plotted this linear and not double log. But again, the growing is linear as expected from the theory, okay? And we were happy that we were able to demonstrate that correlation length increase with size. Several other groups have replicated this result in this and other type of systems. So it is a hard evidence that indeed your brain have not have correlation length of the size of the brain you're considering. So that means that 2 points in the brain are as correlated as you wish. There is no region of the brain that is isolated, thank God. And this is the message for biology, that the brain, being at a critical point, do not need to send extra connections for areas that are far away. Dynamically speaking, changing a single parameter, the brain can be coherent as it wish, the entire brain, to the limit of it being epileptic, or it can be disconnected as it wish, changing a single parameter. Dynamically, this is a very powerful concept. But what about time? From physics, we know that the correlation length, okay, in space grows like n to the alpha. This alpha is an exponent that is only sensitive to boundary condition, symmetries, but everything else, the detail, if you're talking about neuron spins or whatever, are completely irrelevant. The thing is that from the theory, we knew that the length, the distance at which two neurons or regions or spins or whatever are correlated will increase as your observation window or isocysteine size. But these are dynamic quantities. If you measure now the time constant, which time constant usually is used by the engineer to imply the decay to a certain percentage of your observation fluctuation. But in this case, we talk about correlation time because this correlation time is not a finite quantity, but depend on size. And so the correlation length also give you the correlation time, because it's linked to this other exponent. So the only quantity that matters in all of this is the correlation length, because all the others are links. And this is beautiful, because you don't have to be worried about time and space as independent quantities. So, and here you have three things that I mention usually for another audience. This is called dynamic scaling. Is related to what is called critical slowing down, quantities that reaching when the system is near the critical point, things slow down, meaning that if you perturb the system, take more time to come back to the fixed point or to the previous configuration. This doesn't mean that the system slow down it itself. It means that the perturbations have consequence longer in time, meaning that it relax slower, that prevent the system to forget quickly the previous configuration. And here you have the tarantella metaphor. Sorry if I am too metaphorical, but I always explain to the student that if you dance tango, you have 2 degrees of freedom, and if you measure the correlation time of the dance, anyone can make a mistake, stumble, or change the pace very quickly. But if you are in a tarantella, I hope that everybody knows what a tarantella is. It's like a tango with many degrees of freedom, and you are linked to other partners. As the size of the system increases, is more and more difficult to do whatever you want unless you disconnect. Okay, so if you measure the correlation time of a critical tarantella, which means it's not very disordered, it's not very ordered, you will see that as the tarantella increases the number of participants, the time constant of the fluctuation will necessarily will increase. So we decided to test dynamical scaling from data, beautiful data, 10,000 neurons in a behaving mice, meaning a mice that is doing whatever he or she wants, that is being monitored with a technique received the name of optogenetic. Basically, the neurons, when it get activated, change their voltage. They will fluorescence different, and because of this technique, you can image with a TV camera, the brain of this animal as they move around. And then there is an example in panel G. You have 10,000 neurons where the dots represent the firing of individual neurons. On the top signal, you have the animal is running, is in green. The pupil area, how much is open the eyes, because it's also being monitored, is in gray. You will see there are some correlation between behavior and the raster of action potential activity there. But that's not what we are interested. What we are interested is in the global statistic of these correlations during the freely running behaving activity. On the panel A and V, to have an idea, this is like a millimeter square, okay, in the XY axis, and less than a half millimeter going down into the deep layers of the cortex. And you have like 7 layers being monitored individually. So these are technique of 2 photon techniques, such that you can image down into the deep layers of the cortex. Okay, so we compute correlation length on the right side, on the left side. These are different sizes from pink, from violet is a large size, 1,000, so meaning 1 millimeter, and brown circles. I'm talking about this graph that says space. And what is being plotted is the correlation, the connected correlation, cross zero. We're looking for the zero crossing, and then the zero crossing, you see that as I increase the size of my window observation, the correlation length increase. And on the bottom, you have the result of the correlation length in the vertical axis as a function of the window of observation, window size, and each curve represents a layer from layer 1 to layer 11. And you see that as you go deep, no matter the small deviation that are expected from this situation that is not very homogeneous, you see that grows linearly. Okay, so bingo. In the scale of 1 square millimeter, you can show that indeed the cortex of these animals is critical. Now we go and compute the correlation time. Basically, is what in engineering people will call the autocorrelation function, because it's x of t delta t. Now your axis is time from 1 second to 100 seconds. And again, is done for different windows. And as expected, for the short time autocorrelations function values, in the panel V, you see that as you increase the size from 400 to 1000, your tau, your correlation time, increases, and in all the layers, from layer 1 to layer 10. So that you can summarize this, that indeed your your uh, your correlation in space or your correlation in time increase with the size of the system. And there is no other possibility to explain this data unless you invoke that the dynamic of the process is critical. We are not the only one that have measured this. We are the only one that have measured this in the brain. For now, this paper was published a couple of years ago. Is the work of Sabrina Camargo. Is a physicist originally from Sao Paulo that decided to emigrate to the Argentinian scientific community a few years ago, and now is happy with us in the University of San Martin, surviving as everybody else. This work is beautiful in the sense that it was the very first in the brain, but this was actually shown for the first time dynamic scaling by Giorgio Parisi in flocks of birds, together with other researchers, Irene Giardina, Andrea Cavagna, and our truly Argentinian Tomás Griguera. So to show that the brain, that the correlation in time and space are similar to other systems that people have studied before is really encouraging. I'm sorry, I have to read this very quickly. What is the relevance of this? The larger is the system, the slower the time scale. Is the tarantella metaphor. The reason why it's relevant for the brain is that has been observed a certain hierarchy in different areas of the brain. Actually, a researcher from Mexico, Romo, have demonstrated this about 15 years ago in experiments in mice. And it has been very intriguing because it seems that areas that 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 are are are, are supposedly doing very cognitive stuff are slower, like working memory, decision making, that kind of thing. That areas that are trivially receiving sound and processing, and or receiving a sensation from sensory inputs and send it to other areas. So the conjecture in neuroscience is that this hierarchy has something to do with the function, and somehow the brain have evolved to get slower time scales or faster time scale. What we suggest in this paper is that indeed this is for free. The larger the area, if you put more neurons in an area, for whatever reason you put more neurons, you will get slower time scales. And this is something that still is being argued. And actually, we think that is very relevant to understand why we have all the different time scales in our brains. Again, you see that I have trying to persuade you that we are not doing an ad hoc theory for the brain. We have the same solution, mathematically speaking, and we are looking for problems for that solution. Yeah, the problem are maybe in brains, the problem may be in protein, but all my life I've been doing the same thing. I have to confess that. But that's what we are doing. Well, this scaling can be shown at the level of the eigenvalues. I show this just because it's amazing how if you renormalize your axis when you compute the spectra of the eigenvalues of the correlation matrix, and you change your accordingly you renormalize your axis, the rank of your eigenvalues of the correlation matrix, it'll all collapse in a single function. Independently, if you go from 1,000 neurons to 5,000 neurons, indicating that again there is no scale, fine scale, that the system is scale free, which is expected accordingly from the theory. Finally, I told you there will be very quick, only two more slides. I mentioned to you that if you compute your correlation time of your system, you have, independently if it's the brain, if it's the Ising model, if it's a tarantella, you define your order parameter. Your order parameter will fluctuate because it's complex in the complexity jargon, not in the mathematical jargon. So you have wild variability at the critical point, and you measure your autocorrelation function. You know for the theory that the autocorrelation will peak at the critical point, will be maximum at the critical point, will be one actually in an infinite system when you compute for infinite time. This is being plotted here for a numerical simulation of the Ising model at the critical temperature in order the parameters ramp up, one being completely ordered at cold temperature, zero at high temperature. The susceptibility, that is the integral of the variability in black, peaks at the critical point. And if you measure the autocorrelation of the order parameter, is in the bottom, panel B, you will see that have a maximum over there. So, together with the students in the University of San Martin, we say, why wouldn't measure a short time autocorrelation per month function, get the value of the autocorrelation function, and then use a simple feedback loop to change the temperature accordingly. So if the autocorrelation decrease, you will increase the temperature and vice versa. Okay? So it's a very simple-minded control system. And when we did it, we choose a system. In this case, the system is a neural network with the type of network that Haimovici studied in his dissertation I mentioned before, the one that compared the dynamic of the human brain data with the model data. I showed at the beginning of my presentation. We do that model. That model, as I mentioned before, have a single parameter, which is TH, which is the threshold of the nonlinear units. There is no other parameter in the model. You have thousands of neurons interacting according to some adjacency matrix. The adjacency matrix is irrelevant. Is relevant the topology in this audience. The topology have to have some sort of long-range connections, just in order to assure that the system have a critical point. Then you choose the threshold, and if you do it at equilibrium without any control, it will draw this nice panel V, okay, that is plotted there. Now you put the feedback loop, and you start from different threshold in panel E. You start from a threshold very low, and you have the red dots that immediately we put the feedback loop goes to the critical control parameter, bring the system to criticality. Will you put another initial condition for your threshold, a very large threshold, and is the blue line. The blue line will bring the system, will try to maximize the autocorrelation, and it will go into the critical point. The other panels plot the quantities, the autocorrelation in panel D as a function of the threshold, and the dots are the dots for the calculation. You see that go and reach to the maximum for the autocorrelation function, for the critical value of the system at equilibrium, the system automatically will converge to the critical point. Paradoxically, you use a property of slowing down, a property of the correlation, to make the system back to the most unstable point, because the critical point is the most unstable point, the point in which the largest variability, the largest susceptibility, etc., etc. So that was a sort of a trick that we use, not to imply that the brain use this to be always near the critical point, but to show that indeed it is a robust property that can be the correlation, a robust property can be used even to tune the system back to criticality. I reached to the thanks to your generosity of not interrupting and asking very tough questions. I reached to the end. Complexity is very often associated with phase transition. I went through very fast. Some useful language can be used to describe brain dynamics. Multiple reports. I showed some of our reports, but there are a number of other papers that have been replicating this. Some of the papers have more than 1,000 citations at this moment, which make us extremely proud. Our salaries as investigators are increasing proportionate to the citation rate. Brain data exhibits finite size scaling both in time and space. I mentioned quickly that these results of the dynamic scaling maybe are relevant. No, maybe no. Are relevant maybe for the explanation of this intrigues and time scale that neuroscientists are discussing. And the temporal signature of critical design can be used to bring back the system to criticality, which is a sort of a toy model to demonstrate the robustness of these correlations. And I guess that I get to the end. And thanks to all the collaborators across the years, across the planet. And I now shut up. Thank you.
Thank you. So I have one question. So do you just observe or somehow you do some intervention in the brain?
All the data that I show for this presentation are in no perturbation, which is actually need to be defined. The state of the human brain when you are in the scanner that you are recording is that there is no explicit input or output, meaning I don't tell you anything, you don't tell me anything. The condition is called resting state in the generic literature, uh, although it's extremely difficult to define. In one of my initial papers, I have defined it literally in the physical review letter as the dolce far niente state. Italian call it that, to the sweet do nothing. The data from the mice is also on the same condition, freely behaving, but the animal is walking, but it's not actively engaged into any of the behavior, grooming, exploring, or whatever. With other colleagues, we have published papers under sort of quasi isothermic conditions, which means we analyze data from people under anesthesia, going into anesthesia, before and after anesthesia, and we replicate all the results and expected as expected. Under anesthesia, you get the brain gets subcritical, and the correlation length and the correlation time changes accordingly to the theory. In other experiments, we explore sleep, which also is subcritical. In another experiments that were done in collaboration with people in London, we were the first to measure these correlations under a magic mushroom, under psilocybin, together with, at that time, Enzo Tagliasucci was a collaborator in this work. Then Enzo took this psychedelics research further and make much more detailed analysis of the consequence of psychedelics in the dynamics, large-scale dynamics of the brain. And actually, now we are working with colleagues in Hong Kong, analyzing data from mice under the effect of psilocybin, which is the psychedelic drug, which eventually we will stop mentioning as psychedelic drug because this psilocybin is now entering into the pharmacopoeia as one of the treatment for depression, for addiction, and for many things. So answering your question, what I show is the near the equilibrium condition, which are not equilibrium but closer to equilibrium, but there are other work on perturbation, not short perturbation, but quasi-stationary perturbation.
Thank you. Are there any other questions for Dante? Okay, otherwise we will digest all the information that you sent to us with our brains in critical mode.
Okay.
And we will write to you if needed. Okay, thank you very much, everybody.
No, thank you for being so patient. Thank you.